Structure of proteins. Proteins are having three-dimensional structure. Okay. There are a number of factors which determine the shape or the structure of proteins, and the structure and shape can be studied by four different levels. First one is primary structure. Second one, secondary structure. Third one, tertiary structure. And fourth one, quaternary structure. Let us discuss one by one. Okay. First one, primary structure. Primary structure of proteins, which means the sequence in which the amino acids are arranged in a protein. Sequence in which amino acids are arranged in a protein. Okay. Each protein is having a specific sequence. Okay. Specific sequence. If there is any change in any one of the amino acid, will cause drastic change in the properties of amino acid. Drastic change in the properties. For example, hemoglobin. Okay. Hemoglobin contains about 574 amino acid. Okay. If there is any change in any one of this amino acid, will cause defective hemoglobin. Defective hemoglobin. This is seen in patients suffering from sickle cell anemia. Okay. Sickle cell anemia. Person suffering from sickle cell anemia, there will be defective hemoglobin. Okay. Another example is insulin. Insulin contains almost 51 amino acids. Okay. There are two sequences, two polypeptide chains. Okay. One polypeptide chain contains 21 amino acids. Okay. And another polypeptide chain contains 30 amino acids. Okay. These are linked together by disulfide bonds. Okay. Two sulfide, that is disulfide bonds. If there is any change in this sequence, will affect the hormone. Okay. Second level is secondary structure of protein. This gives the manner in which the polypeptide chains are folded. Okay. Or arranged. Polypeptide chains are folded or arranged. Okay. Next point, it gives the shape or conformation of the protein. Okay, shape or conformation of the protein. Okay, this arises due to the folding of the polypeptide bond. Okay, folding of polypeptide bond. Polypeptide bond due to the hydrogen bonding. Due to the hydrogen bonding between CO and NH groups of different peptide bonds. Okay, of different peptide bonds. As a result of this hydrogen bonding, folding of the polypeptide bond causes. Okay, there are two different types of structures, alpha helix and beta pleated sheet structure. Two types of structures are possible due to this folding. Let us discuss one by one. First one is alpha helix structure. This is the alpha helix structure, and this is the common way of arrangement. Here, NH group of one polypeptide chain is bonded to the CO of the another of the adjacent polypeptide chain and gives rise to a helix structure. So, as a result of this hydrogen bonding, the compound, the protein is folded and it gives a structure alpha helix. Okay, here polypeptide chain forms all possible hydrogen bonds by twisting the helix and NH of each amino acid bonded to CO of adjacent atom. Okay, next structure is beta structure or beta pleated sheet structure. Second one, beta pleated sheet structure. Okay, here polypeptide chain is stretched out to the maximum. They stretched out maximum and they are laid side by side and held together by intermolecular hydrogen bond. Okay, held together by intermolecular hydrogen bond, and they are laid side by side. Okay, two different structure, two different polypeptide chains stretched maximum. Okay, and these are laid side by side and held together by intermolecular hydrogen bond, and these are like pleated folds of drapery. Okay, like pleated folds, different pleats, like they are arranged like different pleats, and this is known as beta pleated sheet structure. Okay, third level is tertiary structure. Okay, here this arises due to the folding, coiling and binding. Folding, coiling and binding of polypeptide chains. Okay, and this gives a three-dimensional structure. Okay, this gives a three-dimensional structure. This gives the overall shape of the proteins. Okay, it is obtained by folding and superimposition of secondary structure. Okay, superimposition of secondary structure. It gives two important structures we already discussed, that is fibrous structure and globular structure. Okay, these are the two structures obtained in the case of tertiary structure, the three-dimensional structure. Okay, the main forces which stabilizes these secondary and tertiary structures are hydrogen bonds. There will be hydrogen bonds between polypeptide chains. Then disulfide linkages are there. Disulfide linkages. Then van der Waals forces and electrostatic forces. Van der Waals forces and electrostatic forces. Okay. Due to these different forces, they are arranged in a three-dimensional structure. Okay. Fourth level is quaternary structure. Some proteins are composed of two or more polypeptide chains. Two or more polypeptide chains called subunits. Okay. spatial arrangement of these subunits with respect to each other. Okay, spatial arrangement of these subunits with respect to each other is known as quaternary structure. Okay. Example, hemoglobin. Hemoglobin contains 4 polypeptide chains. Okay, 4 polypeptide chains. Okay, these are linked together covalent, non-covalently. Okay, these are linked together non-covalent. Okay, so this much about the structure of proteins. Okay.